Once we know how much protein is in each of our lysates, we will be working to sort them out. To begin, we will calculate volumes of the liquid lysate that will give us equal amounts of protein in micrograms. Those lysate samples should have approximately the same number of proteins, but it will include thousands of different proteins that are found in the cytosol of bacteria. Eventually, we want to be able to focus on just one protein, beta-galactosidase. To move towards that goal, we are going to separate the proteins in the lysate by their properties. We will be using protein gels to separate out the various size proteins in our lysate. Protein gels are usually run upright, vertically, as in this picture. We will load our lysates into the wells of the gel at the top. Once all the samples are added, we will turn on an electric current with the negative end at the top of the gel and the positive at the bottom. Before we add the proteins to the gel, we will mix them with a detergent called SDS, which is seen right here, and heat them to boil the protein. The combination of heat and detergent unfolds the proteins and coats them all over with negatively charged detergent. This means that the proteins will move through the gel according to their size. Larger proteins will move more slowly in the gel than smaller proteins. Here's an example of a protein gel I found. The authors ran current through the gel and the proteins migrated from the top of the gel towards the bottom. To see the proteins, they added a protein dye. So you can see protein bands in blue. Lane 8 is the standard. In lane 6 and 7 are samples that contains hundreds or thousands of different proteins. One protein is very abundant in both samples, as we can see by the dark wide band. So there's a protein here that's found in great amounts. Other proteins are expressed at moderate levels, which we can see with this protein here, maybe about 65 kilodaltons based on the standard. Other proteins are expressed at very low levels. So we can see tiny bands, and there's faint ones here as well. So the intensity and thickness of the band shows you how much of a particular protein is at a particular molecular weight. Again, we will see something like this in our lysates, lots and lots and lots of different proteins, because we have not done anything to pick out beta-galactosidase yet. In the third part of the lab, we will be using antibodies to specifically identify the protein beta-galactosidase. Our first step will be to transfer the proteins from the gel to a membrane. All the proteins in the lysate will be transferred from the gel to the membrane, not just beta-galactosidase. We need a way to identify a specific protein from all the others in the gel. So imagine that this is the large number of proteins that have been transferred over from the gel onto a membrane. Antibodies allow us to do this. In this picture, we see a red triangle that represents the protein of interest, beta-galactosidase in our case. It is stuck to the membrane. A first antibody binds to the protein. Remember, antibodies are highly specific in their binding. At this point, the membrane will not show any bands since we did not dye all the proteins. We will then add a second antibody, shown here in blue. The blue secondary antibody is specific to binding the primary antibody. Still, there would be no visible band, no way to see the protein or the antibodies. However, secondary antibodies have an enzyme bonded to them. The only place on the membrane where we will find the secondary antibody and therefore this enzyme is the place where the protein is. We then add a substrate for the enzyme. The substrate is colorless to start, but turns a blue-purple color when it is cleaved, or we'd say a bond is broken. This purple color precipitates or forms a solid and leaves the solution, creating a band only in the spot where our protein is located, like this. The more protein present at the position, the more antibody, the more enzyme, the darker and thicker the band. So this would be the results we would see in lab 3, a Western blot.